Madam, my name is Mugilan. My matrix number is 23dbt20f. Today I'm going to carry out one simulation about a staining method. There are two types of stain, which is negative stain and gram stain. So now we're going to go through about negative staining. Negative staining technique is used to determine morphology and cellular arrangement in bacteria that are too delicate to withstand heat fixing. A primary example is the spirochete treponema, which is distorted by the heat fixing of other staining techniques. Can be used to determine the accurate size is crucial. A negative stain can be used because it produces minimal cell shrinkage. The first step, begin with a drop of nigrosine at one end of a clean slide. So it's like, yeah, it's like this. Then the second tip is the essentially add organism and emulsify with a loop. Do not over inoculate and avoid splattering the mixture. Sterilize the loop after emulsifying. So we have to click the loop to transfer organism on the glass slide using this rod. The second step is essentially add organism and emulsify with a loop. Do not emulsify spattering the mixture. Sterilize the loop after emulsifying. The third step is take a second clean slide, place it on the surface of the first slide and draw it back into the drop. When the drop flows across the width of spreader slide and push the spreader slide to the other end. So the fourth step is air dry and observe under microscope. So the air dry and observe under the microscope at 1000x magnification, and we have to see the observation after the last step. Okay, this is the image that we get from the 1000x magnification. Now I'm going to explain about gram staining. Gram stain is differential stain in which a declaration step occurs between the application of two basic stains. This staining method is used to distinguish between gram positive and gram negative cells, allow the determination of cell morphology, size, and arrangement. The first step is making a bacterial smear. Place a small drop of water on a clean slide using inoculating loop. Then essentially add bacteria to the water, mix in the bacteria and spread the drop out. Then flame the loop when done. Allow the smear to air dry. If prepared correctly, the smear should be slightly cloudy. Using a slide holder, pass the smear through the upper part of flame 2 or 3 times. This heat fixes the preparation. Allow the slide to cool, then continue with staining protocol. The second step is staining. Cover the smear with crystal violet stain for 1 minute. Then gently rinse the slide with distilled water. Then cover the smear with grams iodine stain for 1 minute. Then gently rinse the slide with distilled water. Decolorize with 95% ethanol or ethanol acetone by allowing it to trickle down the slide until the runoff is clear. And counterstain with the saffronine stain for 1 minute. Gently blot dry in a tablet of bibulous paper or paper towel. Then observe under microscope at 1000x magnification, and this is the observation. The first slide is the look like this, and this is the second slide. Then thank you.